I'd like to introduce the first sub-theme of today's conference entitled Innovation and Disruption in Care and Treatments. Under this broad topic, we actually have three specific sessions. The first of those sessions is entitled Innovation in Drug Treatment. The second session is Beyond the Drug Innovation. And the session we're about to talk about now is the academic developments and where, in fact, the research is headed. We've pulled together a stellar team of scientists that I'd like to introduce to you now. Dr. John Bell is the director of the Canadian Oncolytic Virus Consortium, supported by a Terry Fox Program project grant, and is also the scientific director of BioCanRx, a network of centers of excellence that aims to bring novel immune-stimulating therapies to cancer patients across Canada. He is a member of the Centre for Cancer Therapeutics at the Ottawa Hospital Cancer Centre, a senior scientist with the Ottawa Hospital Research Institute, and a professor of medicine at the University of Ottawa. Dr. Denis-Claude Roy is a hematologist and a full professor in the Department of Medicine at the University of Montreal. He is also the director of the University Institute for Hematology, Oncology, and Cell Therapy, and also directs the Center of Excellence for Cellular Therapy. Recognized for his expertise in the field of stem cells, Dr. Roy is Executive Director of CellCan, the Canadian Regenerative Medicine and Cellular Therapy Network for Knowledge Mobilization. Dr. Robert Holt, since 2003, Dr. Holt has been a scientist at Canada's Michael Smith Genome Sciences Centre, located at the British Columbia Cancer Research Institute in Vancouver, where he is also scientific co-director of the BC Cancer Immunotherapy program. He also is professor of medical genetics at the University of British Columbia. Dr. Holt's team is recognized for developing next-generation sequencing methods to interrogate the genetics of the adaptive immune system and for identifying novel infectious agents linked to cancer. The moderator for the session is Dr. Stéphanie Michaud, who is also a co-chair of this conference and whom you've met earlier. Dr. Michaud is the president and CEO of BioCanRx, a not-for-profit organization that seeks to accelerate the delivery of innovative immunotherapies from the bench to the bedside. In this position, Stéphanie is responsible for running all facets of the organization, striving to create partnerships between government, not-for-profits, academia, and industry to maximize the impact of research funded by the network. I welcome all of you. Thank you so much for being here, and I'll pass it over to you, Stéphanie.
Thank you so much, Anne-Marie. And first of all, I want to thank each and every one of you for agreeing to participate in this conversation that we're going to have today about the state of cancer research in Canada, what is happening currently, and where you see the research going in the next 5 to 10 years. So I'm really incredibly proud here as the president and CEO of BioCanRx, to recognize all of you as BioCanRx network investigators. Your reputation precedes all of you. You're all incredible thought leaders in your fields of research. And so I'd like to kick it off by asking each and every one of you to identify, what is the main area of research that you're currently working in, and then we'll follow that with a discussion on where you think the research is going, specifically with respect to the development of immunotherapies for the treatment of cancer, and where you think it'll take us in 5 to 10 years. So is there an individual who would like to kick us off? Or I'm going to select one.
I know you're going to select me, Steph, so I might as well just jump on the sword.
I wasn't, John. I wasn't, but there you go. You've stepped right into it. Awesome. Yeah. Thanks.
So, my lab is really interested in the concept of using viruses to treat cancer. and designing and engineering viruses that have the capacity to replicate specifically in tumor cells and not in normal tissues. And the promise of this approach is that we'll create what is really a replicating cancer therapeutic, but it only replicates and grows in a tumor and not in any of your normal tissues. So the dream is that you'll have this therapeutic that will be able to selectively identify cancers, propagate the therapeutic within the cancer, and hopefully lead to better outcomes and less off-target effects. So with chemotherapy, for instance, as Denis-Claude can tell you quite well, there's a lot of off-target effects because it not only attacks the cancer, but also your normal tissues. And so the dream we have with the viruses is to create these little biological machines that will specifically attack the cancer. And I think the new twist that we're seeing is that we now have a whole collection of viruses that have this property. And what we're now doing is what we call arming them, or adding into them genetic information so they're more effective, particularly in stimulating the immune system, so that the patient's immune system begins to recognize its cancer as foreign. So my lab's basically been doing this for many years now, and I think we're really excited to have a few of these viruses in the clinic, and we hope to be able to combine them and not have them just as standalone therapeutics, but combination with other kinds of immunotherapies that you're going to hear about from my two other colleagues in this call today.
And so, John, just to better understand, and for the audience to better understand the combination of, for example, an oncolytic virus with another immunotherapy, this would be a way to extend the reach of immunotherapies for the treatment of cancer?
Yes, that's right, Steph. So the challenge with any cancer therapy is that it may work well in a subset of patients, but what we want to do is make it so they work well in many patients. And so one way to do that is to combine therapies together so that they actually can synergize with each other. And as I said, you'll hear about immune cell therapy, and I think this is one way that viruses can be used is in combination with immune cell therapy so that they can talk to each other and they can help each other grow and attack the cancer.
Great. Thanks so much, John. Denis-Claude, I think you're going to be up next, since you've kind of been introduced by John naming cellular therapies. So if you want to go next, please.
Yeah, thanks very much, Stéphanie. The idea here is that we have been identified in a whole team of researchers a number of antigens that have been found on leukemia cells and other blood cancers, but also that are found on a number of solid tumors. That's mainly the work of one of my colleagues, Claude Perrault, in association with our teams. And what Claude's done is identified a number of peptides, actually small parts of cells that are present on the surface, and they're expressed on these cancer cells. And the idea is that we take somebody else, a donor who's HLA matched, and we can find cells in this individual that are able to recognize these small peptides that are expressed on the surface of the cells, and not only bind to the cancer, but actually kill the cancer cell. So that is very compelling. It's a very interesting approach because it uses cells, and the cells have the ability to be, one, highly specific, and two, to also be able to limit the toxicity. And in a first clinical trial, we have seen very limited toxicity. Actually, only 1 out of 10 patients receiving these cells have encountered side effects, significant side effects that could be controlled, by the way, and is opening the door to treating a lot of disorders. And right now we're starting into another trial where we will, this time, instead of treating the patients, what we want to do is actually to prevent the recurrence of tumor. So which actually would be even better. Like, we would not have to go through a second round of treatment, but we would upfront do a stem cell transplant and add these cells so that the leukemia cells don't come back. And this speaks to the specificity of the of the immune cells to recognize cancer cells. So, and this is we found these antigens on the surface of blood cancer cells, but also they're present on other types of cancers, and it speaks to I think a whole new approach of using immune cells to target cancer, and that's good for many solid tumors.
And those peptides that you're referring to, that these immune cells are targeting, are they unique to the patients, or are they found on a variety of these cancer cells?
Yes, well, they are expressed on the surface of of various cells, and they are specific to the blood cancer cells.
Okay, great. Thank you.
And they could be expressed on other cancer cells also. So if you think of solid tumor, but we can taper and decide which target organ to target.
Okay, thank you, Denis-Claude. And Rob, on to you and the work that you're doing out in British Columbia.
Great. Thanks, Stephanie, for the invitation to join the discussion today. So our team here has overlapping themes with what John Denis-Claude have already described. So the emphasis is on cancer immunology, the same as their groups. And we're taking two main thrusts. One is genetically engineering immune cells in order to better target certain types of cancer. And doing that in a manner that's end-to-end. Right? So not just doing lab-based research, but establishing the infrastructure, at least the minimal infrastructure, to do the genetic engineering, to actually obtain patient cells and modify them, and to administer them in the hospital environment, both here in Vancouver and in Ottawa. So there's a tremendous amount of logistical challenges associated with that. You're very familiar with that, as is everyone. We're all engaged in these sorts of challenges. There's a lot to do. So what we want to do is provide these interventions. We're focusing on CAR T cells. So those are genetically engineered cells that Denis-Claude mentioned, that are proving to be fantastic therapies against certain leukemias and lymphomas. And they work even in patients that have failed all other therapies. They don't work in everyone, and you can relapse after treatment. So as well as providing the therapy initially, we want to understand what is the basis for relapse and how we can improve them. And how could we potentially extend their application beyond blood cancers to other types of solid tumor settings. That's an enormous challenge for the entire field, not just for ourselves, of course. And when that challenge is met, we're going to see an entirely different landscape of cancer immunotherapy. And there's some potential challenges going along with that, such as how do we manufacture these at greater scale? How do we do so in a manner that isn't so prohibitively expensive that they are extensively rationed? So we're not looking just at the biological and scientific aspects of this. We're looking at healthcare implementation research and healthcare economics as well. So that's the main focus in our team. And another focus is cancer vaccines. So there's lots of different types of cancer vaccines. Tumor, because the cells in it are mutated, appear foreign to the immune system. So you can actually enhance the cancer immune response by vaccinating with those mutations. It's a platform called neoantigen vaccine. And then we're also looking at infectious agents in cancer. So it's clear if there's a singular cause of cancer, in the form of an infectious agent like papillomavirus for cervical cancer, if you can vaccinate against that, it's an extremely effective, almost completely effective means of cancer control. So that's a black and white case, and there's been tremendous gains in that area over the past decade. But there's other infectious agents associated with other cancers, like Helicobacter pylori associated with gastric cancer. We work on agents including Fusobacterium nucleatum that's associated with GI cancers, particularly colon cancer, and it potentiates colon cancer development, and it confers treatment resistance. So the hope there is if we can vaccinate against that infectious agent, that will improve the colon cancer situation also.
That is
a lot going on, as you can see.
There's so much going on, and I'm almost fearful of opening up Pandora's box to ask you folks the question about the future and where you see this all going. Of course, all of us from coast to coast internationally have, the world has suffered the impact of the pandemic. We've also learned a great deal about new products and the quick elaboration of new products. And of course, I'm speaking about mRNA vaccines. But I'd like to touch on that and get your thoughts about how that is going to influence, the field of cancer immunotherapy. But Rob, I just wanted to ask a question with respect to the vaccines that you're developing, for example, for the treatment of gastric and attenuating the development of colorectal cancer. How do you find these antigens in a lay summary? Like, how do you develop this vaccine, that's going to be effective against these cancers?
So the classic infectious agents like human papillomavirus were found by purifying the actual agent from tumor tissue. So the modern approach is to do deep DNA sequence analysis of tumor specimens. So you take tumor biopsies and you sequence all the DNA present in that biopsy, and it contains human genetic material, of course, from the patient, but it will also contain DNA from all of the other organisms, the tumor microbiome, present in that sample. So what you do is you use computational methods to remove all of the human DNA sequences, because we can recognize that, because we have a human reference genome to compare to, and then we look at what's left, and we see what's overrepresented in tumor samples compared to healthy tissue biopsies in the vicinity, to other non-cancer patient biopsies. And in some cases, it becomes clear that there's a huge overrepresentation, a tumor infection, you might say, by certain infectious agents. And they can be recognized as pathogens and further characterized. So the vaccination strategy becomes challenging, however. So very briefly, what I mean by that is for HPV, vaccinating against cervical cancer, there's a pre-exposure window before adolescence, where nearly all individuals don't get exposed to the virus, so you can vaccinate preventatively, and that's a very effective approach to vaccination. If you can arm the immune system before it sees the pathogen, you're best positioned. So for many of the other agents, like Helicobacter pylori, like Fusobacterium nucleatum, and many other viruses, you're exposed very early as a neonate, essentially. So you can't vaccinate preventatively. So what we're looking at is strategies where you can vaccinate when a patient is in remission. So they develop cancer, they've been identified as a cancer survivor, and what you really want to do is prevent recurrence. So if we can vaccinate in that scenario to bolster the immune response, we're hoping we can see extended survival. It's going to be an experimental design, but that's an approach that people are leaning towards.
Thank you. Thank you for that. And it's a great segue to the next question about the direction in all of your individual fields of research and what you see coming down the pipe, with respect to your own research programs and where you're taking your research programs. So John, Denis-Claude, Rob, what do you see on the horizon in 5, 10, 15 years? That's going to be a really exciting development, a game-changing development in the area of the development of cancer immunotherapies. What do you see happening?
Well, go ahead, Dave.
Yeah, go ahead, John.
All right. I mean, I think all the people in this call believe in immunotherapy, and we believe in it because, as Denis-Claude mentioned, you actually create a therapy that has a memory. It hangs around. It's a living drug. It can continue to attack the cancer as it changes over time. So cancer is a real problem because it has the ability to change and hide from therapeutics. But our immune systems have evolved to be able to deal with changes. And so they can evolve too. So I think all of us believe that if we can figure out the best way to activate our immune systems and create a situation where the patient's own immune system can deal with the tumor, that's really going to be the game-changing thing. And so you've heard from Rob and Denis-Claude both about ways to engineer immune cells to attack cancer, and I think that's a huge path forward for sure. But I think other ways to think about combining therapies with those immune cells, whether it be a virus or a vaccine or other things, that seems to me be the direction that we're headed, just finding better ways to help the patient's own immune system identify that the cancer is a foreign entity and keep it from recurring, which of course is another huge value. And I think the only other thing I'd say before I let you go ahead is, I think for us, we like to see that these immunotherapies are applied earlier in the patient's treatment journey, as opposed to at the end of the journey, because we know that some of our other kinds of therapies damage your immune system. So I think if we can start to move these therapies forward in the treatment of a cancer, we have a better chance of them being successful.
Yeah, and we've certainly seen that this year in some clinical trial results that have come out of the U.S. Denis-Claude.
Yeah, I wanted to add that the scientific developments to propel our research. Like only 10 years ago, we couldn't have done what we have done right now. And this is allowing us to identify new targets. We have new strategies to get the immune cells more efficient, more active at killing the cancer cells, at getting selective activity. And what I foresee is soon we will have a library, or you'll go to the pharmacy and you ask for this drug, and it's going to be provided you in a vial, and then you inject the cells and they're going to do the job. Maybe I'm simplifying just a little bit.
It sounds very exciting.
But I think that improving on targets and increasing accessibility, making cells that are active against cancer, but they can use it in many individuals, like Rob's cells or John's viruses, is like to take that off the shelf, you inject that into patient. And hospital pharmacies are going to be able to have access to these novel medications. I'm very enthusiastic.
You are very enthusiastic, and I'm going to park my follow-up question because I want to hear Rob's thoughts about the future. But I'm really curious in, for what I've heard from John and from you, Denis-Claude, is we're going to have lots of options. And, of course, my question is, how are we going to manage that? How are we going to afford that? How are we going to support the research to develop all of these options? But, before we get there, Rob, your view of the future and where it's going.
I'd certainly agree with the other panelists. I think an important paradigm is going to be combination therapies. So we're currently in a situation where often one cancer drug, even a highly targeted drug that can be very effective, one is tried at a time. Often there's a response and a relapse, and then another drug is introduced, another treatment type, like an immunotherapy. Eventually, if patients fail all standard therapies, they get new experimental ones, sometimes which work fantastically. Some of them have recently been introduced into the clinic as more standard care, like CAR-T. But I think what's needed is a more systematic evaluation of what combination therapies are most effective and introduce them into standard care early. Even though they will be more expensive at the outset, I think we'll see more durable responses and even cures. If we take that approach, it's analogous to HIV therapy, right? In the early 80s, there was no treatment for HIV. And what turned out in the end to be effective was highly active antiretroviral therapy, which is combination therapy. So you target different molecules within the virus and the virus replication cycle simultaneously, so it doesn't stand a chance to propagate from day one that you introduce the therapy. So that has been explored, of course, with conventional chemotherapies. You rarely get a conventional chemotherapy on its own these days. They're combination therapies, but it hasn't yet been explored systematically for targeted therapies or immunotherapies. So that's the,
yes.
But then they do become expensive, right? So that has to be done, on a backdrop of equitability. So how are we actually going to be able to afford these therapies if they turn out to work well? Which some of them will.
Which some of them will, and yes, absolutely, and we're already seeing that, now in our current healthcare system and the challenges, with some of the cellular therapies that have been approved. For example, back for example, to include of the pharmacy, where you just go, you get your vial, and you're able to, potentially self-inject. How are we going to get to determine, what we need in our science and our preclinical or clinical science to be able to figure out what are those combinations, how are those combinations going to work, how are we going to bring this forward? How would you map out the process in order to be able to achieve and really tailor these treatments in the way that you're describing?
Yeah, I think that one of the things to do is do clinical trial. We're going to need to develop first the basic and basis of doing these treatments in the lab. And once we have developed a rationale and we know exactly how it works, then we can implement it into humans and do it. And basically get the clinical results. I think we will need to devise new clinical trial strategies that address different types of patient population, maybe smaller numbers of patients, maybe compare one combination with another. But this all involves funds. So you're asking about the feasibility. So this means that we need more funding. I think the elephant in the room is that cell therapy right now is very expensive. I think you alluded to that earlier, Stéphanie.
Yes.
We need to be able to afford that. But if you're giving the treatment early and you prevent treatment, the disease recurrence, you prevent recurrence of treatment, and you're not only saving lives, but you're saving money to the system. and you're improving patient health. So we need to intervene very early and be active, and in the end, these treatments are important.
I really want to pick up on what you just said, Denis-Claude, about bringing these treatments and treating patients. All of you have mentioned this about moving up the utilization of very effective therapies like immunotherapies, forward instead of administering them at a later stage of treatment, which is all fine and good, but how are we going to be able to to pay for all of these treatments that you're seeing down the pipe? How are we going to be able to afford to be able to treat these patients with these therapies that for the most part are all biologics? What do you have in your toolbox to try to make these more universally accessible to patients across Canada?
Well,
I think that as we go, maybe I'll let the others comment, but very quickly, I think also that as we are developing this, we're taking that the costs are going to bring that down.
Yeah, I think that's
John, you wanted to say.
Right. Yeah, I think it's always true that they cost more at the beginning, but they can go down a lot, and I'm hopeful that'll be the case. But I think another point is, as a country, we have to do more to support research overall, because that's clearly the answer to so many of our issues in terms of economics and so on, is to put more investment into research of all kinds. Obviously, I'm biased towards cancer research, but if you look, Steph, over the last 3 years of the pandemic, I don't know what the final number is going to be for the Canadian government to invest in COVID research, but I'm sure it's a couple $2 billion overall. And that's been necessary for us to stop the pandemic and prepare for the next one. And, 40,000 Canadians will have died of COVID over 3 years, which is a terrible tragedy. But, cancer is 80,000 every year, and it's going to be ongoing. So, we need to be more realistic about what it takes to deal with this very complex disease and invest more money into cancer research so that we can do the kinds of studies Denis-Claude's talking about. You need to have resources to do these clinical trials to find out what's going to work for patients and what's not. And without more support, we're just not going to get there. I was just reading today that Canada is still investing about $8 billion to support the fossil fuel industry in our country. So we talk out of one side of our mouth saying, the fossil fuel thing's a problem, we've got to stop doing it, but we're still putting $8.6 billion into it, and we're putting very little, relatively speaking, into cancer research, which is a huge problem. So I just think we have to, as Canadians, decide to prioritize our health, and the way to prioritize our health is to get good research to come up with new therapies that will be cost-effective and save lives.
I couldn't agree more, John. I couldn't agree
So
more.
Sorry, go ahead.
It seems like there is an enormous amount of investment in cancer research, and there is, right? It's $500 million per year.
$500 million a year, according to the CCRA. Yes, about. It goes up, it goes down, but about on average 500 million a year.
So more investments needed. Right? So the only way we're going to make advances against cancer is through research. It's not going to go away on its own, obviously. And just to put that in perspective, so that was the Canadian Cancer Alliance report, right?
Mm-hmm.
Canadian
That's
Cancer
right.
Alliance, recently
Yes.
published. So they've also published reports that put the total economic burden of cancer in Canada through the direct healthcare costs and the fully burdened costs, including lost earnings, etc., at $26 billion per year.
That's right. That came out this for 2021. Yes. Mm-hmm.
That means the investment in research is 2%. of the fully burdened cost. So that is inadequate. And what it does, in fact, if we don't have that public sector support to do the research that all three of us have just described and address the priorities that all three of us have just noted, and I don't think are controversial. I think the community at large would agree with the things that we've noted so far. It's inadequate, and it means that it's essentially left to industry, and the driver becomes profit, not health improvement. So we need both public sector and as much philanthropic investment as possible so we can run academic, self-sponsored clinical trials and do the preclinical studies necessary to support those trials in order to make the advances that are needed.
A question for you, Rob, and for you, John, Denis-Claude. On the funding, you've made a very compelling case, Rob, right, in citing those numbers about the cost of cancer to society and relative to the amount that we invest, which is jarring to say the least. What role can patients, other than as participants in clinical trials, what role can patient groups play in being part of this team, right, in order to make this research advance so that we can derive as much benefit as possible from the investments that we're making in research in Canada? What role can these groups play?
They have to be advocates. They have to advocate for their disease, for research, for development of new treatment. And industry has been asking the academia to developing the new treatment. So, and clearly advocate not only the benefit of the patient, but also there's creating an economic drive, right? It's positioning Canada as a world leader in new treatment, and that has economic value.
I couldn't agree more. So on that subject, Denis-Claude, the Government of Canada in last year released a life science strategy. It's called the Biomanufacturing and Life Science Strategy, investing about $2 billion in total in this strategy. From all of you here, you're all very, very active researchers in your field. You know what it takes to make things move forward. You also know what it costs, and you also know, all of the human resources that are needed writ large and the support needed writ large to advance, your research forward and to get it to the clinic. What's your perspective on what the impact of this strategy will be on your work and on advancing the field? Is it enough?
Well, I think it's a great start. I think that, considering that we were not investing in this activity at all before, and now what the government's recognized is we haven't had the infrastructure to advance our discoveries to the lab, to the clinic, and so they want to put a bolus of cash in, and that's great, but it's the beginning because, Denis-Claude has a manufacturing facility he has to make sure is viable, and when people work there every year, they don't just work there 1 year, and so we have to have consistent funding, a stream of some sort to maintain these facilities so they can actually become the world-class organizations we want and maintain themselves as well, which in the end, they will become self-sufficient, but we really have to have some consistent ongoing funding. It can't just be build a nice building and then walk away from it. You have to actually build a building, train the people, employ the people, and keep this all going. So I think I really appreciate the $2 billion initial bolus, but that has to be just the first, and we have to have more ongoing. As Rob mentioned, the amount that goes into cancer research is quite small compared to the cost of society, and $2 billion is still quite small. If it's only one time, it's minuscule. We have to have a consistent funding stream to keep these activities going.
Yeah,
Yeah,
it's
we,
a good
we
start.
need to be able to go further. And with that, we
Absolutely.
need additional fundings. And biomanufacturing is a way of being independent, and pushing in that direction.
Yeah, definitely. It's it's all over the documentation. This is about promoting our self-reliance. But what happens sometimes is that, all of you folks know, John has just mentioned it. We need sustained funding over a much longer period of time if we're really going to move the needle and become sustainable in our biomanufacturing activities, facilities, et cetera, right? So that we get this engine going and it's producing and it's treating patients, producing products, and there's an ebb and a flow to it. Rob, any comments on that with respect to the strategy or anything else?
Yeah, no, it's very welcome, right? We are kind of playing catch up, so it's fantastic to see the investment beginning. There are a couple things I think could strengthen the strategy. Those include the ability to assess and reallocate over time, depending on what is being effective and what less so. So it needs to be a nimble strategy. And I'd really like to see investment not just in building something, but in buying something. So what I mean by that is we're seeing billions of dollars now going to both academia and industry for this infrastructure, which is great, but it is uncertain once it's built. What tech transfer will occur to begin to manufacture products, and then when those products are made, what is the market? There's a lot of market uncertainty. Who's going to buy them?
Mm-hmm.
Well, there's an
Yes.
now with COVID vaccines, but the market's actually, at least in our country and several other developed countries, saturated for COVID vaccines. So it's maybe not the best long-term plan. And the great uncertainty, the great chicken and egg challenge, a lot of biologics and cellular therapies is that there's such an enormous demand and resources to make them, and then an uncertain market is going to purchase them, yet patients need them. So what would be fantastic if our provincial ministries of health who reimburse for drugs made a commitment to actually buy the products that are going to be manufactured for Canadians. That would
As
kind
a
of close
result,
the circle. Yeah.
I agreed, absolutely. And we'd be able to achieve that sustainability, right? And there would be, just like you've said it, market certainty, which goes a very, very, very long way. Gentlemen, I think... Yes, go ahead.
One more point I can't leave out is our trainees, right? So
agreed. Yes, we cannot.
Is done by trainees, and they are under-supported, drastically under-supported, and it's becoming difficult to recruit. They need more support.
Couldn't agree more. Couldn't agree more. And I know that's very top of mind right now for government, and I know a lot of folks are speaking to government about the support of HQP, and that it's a necessity. We absolutely must do more because the amounts that they're receiving are not adequate. Gentlemen, I think I want to sincerely thank you for sharing your time to have this conversation about the work that you're currently doing. That's so exciting, so profound, what your vision for the future is, and also really appreciate the role, right, that you play in the ecosystem, as researchers, of course, but really leaders in the area of science in Canada. And so for that, I really thank you. It's an honor to know you all. So thank you for your time.
Thanks, Stephanie.
Thank you.
And gentlemen, on behalf of CCC, thank you so much for your contributions today. And Stephanie, as always, for your leadership in co-chairing this conference, and of course for your magnificence in maneuvering a very large conversation that I think we could probably have a full 2-day conference on in and of itself. So we have a number of questions from the audience, and we only have about 10 minutes for that, so we're going to bridge into those right now. We welcome back Denis-Claude and Robert for a Q&A session. They've graciously agreed to be here today. Thank you so much for that. We have at least so far half a dozen questions that have come through, and so I'm going to take them in the order in which they have been shared with us. There was in various different conversations that each of you brought to the table this concept of bringing patients into earlier stage processes. In other words, putting them in sooner. So there's a number of questions actually related to how, in fact, do you see the engagement of patients and, frankly, patient groups in your research? Denis-Claude, maybe I'll start with you.
Sure. Thanks, Carol. Well, there are many ways I think patients can be involved. I think the first thing that comes to mind is actually within protocol design, when we are deciding on how to treat patients and deciding what's the endpoint, how are we going to do things. Clearly for patients to be involved. And maybe I can give a very short example where sometimes you have physicians who think it should be done in a certain way, and then there are patients who think you might think differently. And in one of the clinical trials, the physicians were thinking, for example, that tapering of the immune suppression in patients who had a transplant, whether it's a kidney or a stem cell transplant, was less important than treating the disease and or making sure that the graft was doing well, while the patients felt that the medication was giving them so many side effects that actually it was a priority for them. So as soon as we would control their disease, they would be ready to start tapering, whereas the physicians thought, well, maybe we should not do that and aim at reaching out a longer duration of control and things like that. So I think we have to strike a balance. It's not clear that one necessarily has a definite opinion, but even I've had more decisive points where patients advocated for something and physicians advocated for another. And clearly the patients are the one for whom we're doing this. So we have to listen to them.
And may I ask, Denis-Claude, is this part of your process today? Are you actively engaging patients in your own research in your labs?
It was not the habit to do that, but actually, since the experience I've had with these patient engagements and discussions, we are engaging patients into this. I think one of the difficulty is selecting the best patient to answer these questions, those that know the area and want to be involved, and also it's a question of time and energy. But clearly, I think patients have a pace to be involved, and we want them initially.
Yeah, thank you for that. And I might add that patient groups can actually be supportive of helping you to find the right patients for your work as well. Rob,
a quick thing is advocating. I think I won't expand on that, but clearly being advocates of nutrients, I'm sure Rob will agree with me that we need them to back us researchers and clinicians who want to develop new treatments. So.
Thank you. Rob, I'm going to ask you the same question. I'd love your perspective on it as well. What it is specifically you're doing at your end.
Yeah, thanks, Carol. And I absolutely agree with Denis-Claude. So we work with patient advocates and patient groups on several of our projects, and I think it's fantastic. So there's certainly aspects of clinical trial design, future directions, and lobbying and advocacy that absolutely they're helpful pushing forward. At a more fundamental level, we often have some of our patient advocates join our regular scientific meetings. So this is at the earliest stages of development and throwing around new concepts and successes and failures in the laboratory. So I think it's helpful for both groups, so us as scientists, but also for the patients to see that science, for them, I think it's helpful. They've said it's helpful to see how messy science actually is, and how unclear the goals and the steps towards them often are. And then on our side, from the scientist side, it's extremely helpful to remain grounded. So we otherwise at our internal meetings tend to just geek out and talk about sciencey stuff that either is not going to be practical and helpful in the long term, or it's so embedded with jargon and scientific language that is completely incomprehensible, often even to other scientists in the room. So it makes us communicate more clearly and be more rational. So I think it's really a win situation.
Thank you both for being trailblazers in the space, and we'll look forward to engaging you in some of the work that we're doing on another initiative. I'm going to go directly to read a question from the question system here. The question is, immunotherapy has shown incredible benefits to a subset of patients with cancer, specifically in colorectal cancer, only about 5% of tumors. How far has the research come in terms of making a greater percentage of unresponsive tumors more responsive to it?
Yeah.
I'll give the question to either of you.
Go ahead, Denis-Claude.
Yeah, okay. Well, actually, it's an interesting question. Everybody's tackling this in different ways. I think immunotherapy right now is working a lot into in vivo, like injecting medication, but we're also developing cells that will be targeting cancer. So actually, I know several groups, even here in Canada, that are working on this. It's an objective internationally, and it's hard to put a number on how this is going to develop, but clearly there is a path forward. There are antigens that are being found on these tumors. There are people preparing to tackle this, whether it's TIL cells or CAR T cells, and we've been actually treating CAR T cells for people with sarcomas, and solid tumor is a field that's developing in solid cancer. So GI cancer is certainly one that's a prime target.
Rob, any further comments?
Sure. I would point to the success in CAR T therapy for hematological cancers as being quite different from the track record of success in solid tumors, the most common cancers, which I think is the basis of that question. So the common cancers are more challenging for all sorts of different reasons. So, when an immunotherapy is successful, it's successful in one way at killing the cells, but if it's not successful, it can fail in dozens to hundreds of different ways. So it's kind of a matter of defining all of the failure modes and try to find workarounds for them. So they can be quite different, right? As solid cancer, like colorectal cancer, can be immunoresistant because it doesn't express appropriate foreign antigens that immune cells can recognize. Or if it expresses them initially, they might subsequently be lost. The immune cells themselves can be exhausted and ineffective and need rejuvenation. The tumor cells back on that side can express and secrete immunosuppressive factors, many different kinds. So it's a very complex situation in the solid tumor environment. And it's a matter of identifying what all those complex mechanisms are, finding out which is responsible for lack of response in an individual patient or small group of similar patients, and then finding effective workarounds. So it's challenging, but things are advancing one small step at a time in that realm, for sure.
Thank you. I'm going to finish on one final question. We only have a couple of seconds, but I feel like I would be remiss if I didn't ask this question with the two experts at the table. How far are we away from a cancer vaccine? There's been so much press about this in recent times. Just quick answers from both of you, if I could.
I think maybe a quick answer is technology is coming to fruition. I think COVID has taught us that mRNA vaccines are available. And therefore, this is a tool that is being targeted for solid cancer, for cancer vaccination. And there are antigens that, as I mentioned previously, that are being identified as we speak. A number of them have already been identified on tumor cells. So the vaccine is not a fancy idea or dream anymore. I think it will come to application. There will be many questions raised onto which patient, which antigen, which tumor, at which time, before the cancer, after the cancer. Usually it's going to be, the first vaccines are going to be after a tumor, because we want to make sure that these patients have the problem, and that there is a solution. We need to find solution for a problem before tackling the whole patient population, which may not be affected by this cancer and which may never encounter this situation. So actually, there are many issues that will have to be solved, but clearly there's a patient population, there are antigens, and there's a methodology. So it's a question of time, but I would say we will see this, and I would say I rarely want to venture in years, but I would say 5 to 10 years will have. And before 5 years, we'll have them in clinical trials for sure.
My goodness, thank you. Rob, I'm going to give you the final brief words, if you don't mind.
Sure.
I know
Things are very encouraging, right? So we already have an effective cancer vaccine, as I mentioned earlier in the panel, against HPV, for example. So that's a fantastic success story. On the other end of the spectrum, we actually might never get to a universal cancer vaccine. So what's going to happen in the near term is, I think we'll be able to develop effective vaccines, either preventative and or therapeutic vaccine interventions against specific cancer subtypes. And the more of those that we can intervene against, the better, and things will gradually improve.
Thank you both. I wish you'd had more time for our Q&A. We very much appreciate you being here. And thank you again for covering with Stéphanie and John as well, a lot of very important territory. We'll see you again.
Thank you.
Thank you, Carol. Thanks, everyone.